Our next speaker today is Joel Solar. Did I say that correctly? Soler, sorry. Joel is a fourth-year graduate student in the Behavioral Neuroscience program at MSU. Prior to initiating graduate studies here, he graduated summa cum laude from the University of Puerto Rico in 2014, where he majored in experimental psychology with a chemistry minor and also held a swimming scholarship. Upon arriving at MSU, Joel entered Dr. Lily Jan's laboratory in the Department of Psychology and initiated projects that focused on examining the role of environmental lighting on learning and memory. Since starting his scientific research career here, Joel has received various awards from professional societies and federal funding agencies, including an Academic Achievement Graduate Assistantship from the Graduate School here at MSU, an honorable mention from the Graduate Research Fellowship Program from the National Science Foundation, and a Diversity Training Supplement Grant from the National Institute of Neurological Disease and Stroke, NINDS. Most recently, Joel has published a paper with his advisor, Dr. Jan, who is here today, and two other faculty members that has gone on to receive coverage in over 40 national and international media outlets. And I believe that research is on the development of the hippocampus in low lighting conditions. Is that correct? Okay, so welcome, Joel.
Good morning, everyone. It's an honor for me to be able to talk to you today about my research efforts into understanding how light modulates learning and memory. So, as many of you probably already know, environmental light intensity fluctuates across the seasons, with spring and summer months displaying enhanced lighting intensity, while low levels of light are predominant during the fall and winter months. In addition to this, there's a positive correlation between light and learning and memory performance, with bright light yielding better performance than dim lighting conditions in humans. And there's been an extensive line of research studies that have documented the beneficial effects of bright light in a wide variety of human populations. Long-term bright lighting has shown to improve academic performance in elementary school children, increase working productivity in adult office employees, and even slow down cognitive decline in elderly dementia patients. However, despite the abundant information of how light impacts cognitive function in humans, the exact neural mechanisms are not well understood due to the lack of a suitable animal model, because traditional rodent models are nocturnal, and the onset of light induces a slowing down of general activity to the point of even inducing sleep in these animals. And in humans, the onset of light promotes wakefulness and arousal. And that's where the unstriped Nile grass rat comes into play. This rat hails from Kenya, and it proves to be a suitable animal model due to its diurnal activity pattern resembling that of humans. These animals were brought in the late 1990s here to MSU via Drs. Kay Holekamp and Laura Smell, and currently MSU has the only well-established breeding colony in all of North America. And we plan to utilize this animal model to look at how light affects the brain regarding learning and memory performance. So our lab has established a chronic daytime light deficiency paradigm in which we will assess how chronic dim lighting conditions affect learning and memory. In this setup, one group of animals will be housed in 12 hours of bright light and 12 hours of darkness, which mimics a summer light condition. In the other group, we put them in 12 hours of dim light and 12 hours of darkness, which mimics a winter light condition. And the animals will be housed in their respective paradigms for 4 weeks prior to behavioral training and testing. So once they've undergone those lighting conditions, we then assess their spatial learning and memory with a task known as the Morse water maze. The goal of this task is for the animal to locate a submerged platform in a pool utilizing external visual cues. If the animal has learned well, it should spend the majority of its time searching for the platform in the immediate vicinity once it's removed. And here we have an outline of the pool with the light gray region representing where the platform used to be, so we denominate this as the goal quadrant. In the left-hand side, we can see that animals in the bright lighting condition search for the platform in the goal quadrant more effectively than their dim lit counterparts over here on the right. To be more precise, animals in the bright light conditions spend approximately twice the amount of time searching for the platform in the gold quadrant in comparison to the dim group. In addition to behavior, we also want to look at a brain region that is essential for learning and memory, known as the hippocampus. It is this red highlighted region that you see in the first image. And specifically, we wanted to look at neuronal communication in this brain region because it is essential for learning and memory. In the below diagram, there's a diagram showing how neurons communicate with each other. And what we found is that there is a higher number of circular protrusions that are responsible for neuronal communication sites. And in the below image, we see that in the dim lit group, there's approximately a 30% overall reduction of these neuronal communication sites that are responsible for the learning and memory deficits that you've seen in the previous slide. So we conclude that chronic daytime light deficiency impairs spatial learning and memory by reducing the neuronal communication between hippocampal neurons. So what's the grand scheme of this? Well, according to the EPA, the average person spends approximately 90% of their time indoors, which inherently reduces their exposure to bright light. And this can have a negative impact in our brains, especially within the developing brains of children and the aging brains in our elders. Our research is aimed to spark the development of novel therapeutic strategies to reduce this occurrence. And with that, I'd like to thank my advisor, Dr. Lily Yan, for guidance and support, the graduate school here at Michigan State University, and the NIH for financial support. And I'd be happy to answer any questions. Thank you for your attention.
Thanks.
How did you determine, oh, go ahead, did I hear a question
I
down
just
there?
wanted the light.
Oh, she wanted the bright light on. How did we determine that the traditional rodent was not a good model for this, and that this particular one that you found down in Kenya would be the right rodent?
So most rodents that are used in labs are nocturnal by nature, meaning that usually they're more active during the night. And if we want an animal model that can translate to humans, we would need an animal model whose activity pattern resembles us.
Okay.
Well, thank you for your attention.
Anybody else? Anything else? This was terrific, and thank you very much for
Thank you.
honoring us with your presence today.